Land capability classes. Determining the best use for a piece of land is more and more important as the population of the planet increases, but the size of the planet does not. Developing countries need to choose crops with care so that they can successfully feed an increasing population and cultivate stability. And we need to practice soil and water conservation so that we preserve all types of land for future use. Land capability factors such as internal drainage and soil depth determine the best crop use for a piece of land. Land capability classification is the process of assigning a class number to a piece of land based on its capability factors. There are 8 class numbers designated by Roman numerals. Class 1 is the best land in terms of arability, or a land's ability to produce row crops, such as corn, wheat, potatoes, sorghum, etc. It has almost no slope, deep soil, good internal drainage and structure, and good surface drainage. The soil is loose, easy to manage, and does not require special treatment to control erosion and runoff after it has been cropped. Most crops can be grown on Class 1 land, so it's the standard to which other classes of land are compared. Slope increases through land classes 1 to 4, which are grouped together as land that can be cultivated for row crops if soil management techniques such as terracing, strip cropping, and contour plowing are used. Land classes 5 to 8 are not recommended for row crop cultivation for reasons including rocky outcroppings, problems with flooding, tendency to erode, and shallow soil depth. These lands are used for pasture and to cultivate trees and shrubs. Class 8 land is the least suited for cultivation as it is generally covered with water and useful for waterfowl habitat. And now, let's explore the core. From the flats of Arkansas to the terraced hillsides of China, rice is a crop produced and consumed all over the world. 1/5 of all calories consumed by humans come from eating rice. Golden rice is a genetically modified rice crop developed for consumption in areas where food is deficient in dietary vitamin A. Golden rice is loaded with beta-carotene, which is broken down in the small intestine to form retinol, a form of vitamin A important to vision. According to the World Health Organization, dietary vitamin A deficiency causes 250,000 to 500,000 children to go blind each year. So how do scientists genetically modify a crop like rice? Remember that DNA, or deoxyribonucleic acid, is found in the nucleus of cells. It contains information for making all the proteins that make up the rice plant. The shape of DNA is described as a double helix or a twisted ladder. DNA is made up of building blocks called nucleotides. Each nucleotide is made up of a simple sugar, a phosphate, and a nitrogen base. The simple sugar is deoxyribose and gives DNA its name. There are 4 nitrogenous bases that make up the 4 possible nucleotides: adenine, guanine, cytosine, and thymine. All organisms have DNA made up of these 4 nucleotides. The phosphate of one nucleotide bonds to the sugar of the next nucleotide, so that bonded nucleotides form the long chains down the sides of the ladder. The 2 chains of nucleotides come together in specific ways to form the ladder rungs. Adenine always pairs with thymine, and cytosine always pairs with guanine. These are referred to as base pairs. If all organisms have the same 4 nucleotides in their DNA, then what makes rice somewhat different from other grasses and considerably different from a cow? The differences come because nucleotides are arranged in different sequences along the DNA strands of different organisms. The sequence TTT-GACT carries a totally different genetic message from the sequence AGC-CTTC. One gene can contain about 1,200 of these base pairs, which allows for lots of differences in the coding of each gene. Rice was the first crop plant to have its genetic code, or genome, sequenced, and scientists found that it contains approximately 37,544 genes on its 12 chromosomes. To create golden rice, scientists use recombinant DNA technology to insert genes from a daffodil and bacterium into the rice genome. Scientists first used special enzymes to cut or cleave a DNA fragment from the daffodil that codes for the production of beta-carotene. The key to recombinant DNA is that enzymes used to cut the double-stranded fragment form short, single-stranded ends, called sticky ends. When DNA from both the daffodil and the bacterium are cut with the same enzyme, their sticky ends will pair up. The daffodil DNA fragment containing the beta-carotene gene can be recombined in the bacterial DNA. The bacterium serves as a carrier, or vector, and will carry the beta-carotene code into the rice cell. When inserted into the rice cell, the vector that contains the daffodil gene will enter the rice cell's nucleus, and the rice cell will begin to produce and accumulate beta-carotene. The golden color of the rice is an indicator of the abundance of beta-carotene, a nutrient found naturally in orange vegetables such as carrots and sweet potatoes, and in some golden flowers like daffodils. Today's SAT word is deficient. Deficient is an adjective that means lacking an essential element or not having a proper amount. In the lesson, we learned that golden rice was developed to feed people in areas where the regular diet is deficient or lacking in beta-carotene, a nutrient essential to vision.